Welcome back, everybody, and we're going to start right up on the slide that we stopped on in our last video. So in this question, I'm asking you to tell me what the D time is. In the scenario, you have to sterilize some item, and you're told that the D80 is 9 minutes. So decimal reduction time at 80 degrees Celsius is 9 minutes for a given amount of bacteria, okay? The amount of bacteria present in the sample is 32,456,200. So what is the D time? How many D times would you have to use to kill the bacteria? How many minutes does that mean it would take? And what is the temperature? So the first question is, what is the D time? I told you the D time right here. So the D time, D at 80 degrees Celsius, is 9 minutes. So that's your D time. How many D times would it take to kill off the 32,456,200? Well, for that, you have to know how many times. So for each D time, you're killing off 90% of the population. And so here's that decimal point. You're going to move it over one time for the first 9 minutes. Then you move it over a second time for the second D time, third, fourth, fifth, sixth, seventh, and then 8. So it would take 8 D times to kill off these bacteria. So if it takes 8 D times and each D time is 9 minutes, the number of minutes would be 8 times 9, which is 72. And then what is the temperature that we're using? It would be this number right here, 80 degrees Celsius. And so if this is confusing to you, you need to come to one of my virtual office hours and we can talk about it, because I can definitely go over it with you. Usually this is a lot more understandable than the growth rate, that equation, but some people do struggle with this. So definitely, come talk to me if you're struggling with this, okay? So now let's look at how we can use temperature to control microbial growth. Heat is very widely used, and it's in general really effective. We use in our laboratory almost every day dry heat sterilization when we are sterilizing our loops, when we're sterilizing our needles, when we are running the test tube lips over the heat so that we don't get any contamination. Moist heat is also very effective at killing pathogens. In fact, it often is better at killing pathogens or at killing bacteria. And so we use moist heat in multiple mechanisms. So moist heat would be, an example would be an autoclave. I use an autoclave to kill off billions and billions and billions of cells. When I am killing off all the biohazard media that you have produced during the labs. Then we can use refrigeration, which is the opposite side. Now we're cooling everything down. Refrigeration slows microbial growth and sometimes can stop it. And then freezing stops microbial growth in general, and it can sometimes kill pathogens, not always. So pasteurization is a form of moist heat, and what it does is it kills pathogens and reduces spoilage microbes to the point where it's deemed safe. There are 2 types of pasteurization that we typically look at: high temperature short time, HTST. And this is what we use when we are pasteurizing milk and other juices that sit in the refrigerator. And then you bring them home and they last for a few weeks in your refrigerator. But there's another mechanism called ultra high temperature pasteurization, which can be used to pasteurize milk for a super long term. And the storage then can be without a refrigerator. So in that case, you heat the temperature up extremely high, and then you drop it rapidly. And so this allows you to store milk and other items in on a shelf. And so it has a longer shelf life versus in a refrigerator. Another mechanism which I talked about is sterilization with pressure. And so an autoclave uses a very high temperature, but it also uses pressure to increase the temperature even higher. So we have typical temperature boiling is 100 degrees Celsius, but when you increase the pressure in the autoclave, it increases the temperature. And so we just increase the pressure, causing the temperature to increase as well. And so what that does is it allows us to get to a temperature of 121 degrees Celsius or 250 degrees Fahrenheit. And we can leave it there for 15 minutes, and it typically will kill all life. If we have a larger volume, we turn the time up to about 30 minutes. And we can also raise the temperature up if we want to make sure that we are sterilizing everything. We can use high pressure processing. So in this case, we're using a pressure chamber to actually kill microbes. This is often done in factories when you're producing cans. Use these high pressure processing to make cans of vegetables that aren't going to have any problems. Like back in the 70s and 80s, if we had a can that was dented, we weren't allowed to use it. We had to put it back because it could potentially be or contain botulinum toxin. With high pressure processing now, we can use those dented cans. There's going to be no potential for any endospores to be in those foods. We can also use desiccation. So desiccation is going to be taking all of the water out of an environment, or taking the majority of water out. So you're basically dehydrating the material. Oftentimes this is with the use of either sugar or salt. So think about your beef jerky. That's what I usually think about. Beef jerky is preserved meat with the addition of salt. So it's been desiccated. And so organisms don't grow effectively because there's not enough of a watery environment, and so they're going to dehydrate if they're on that food. Lyophilization then is desiccation along with freezing. And so this can kill microbes or can store materials for long periods of time. Ionizing radiation then is a mechanism where we use gamma rays. Gamma rays destroy DNA and they damage the plasma membrane. And so this is a mechanism to sterilize heat-sensitive materials. If we use non-ionizing radiation, it is useful for surface sterilization, but it can't actually penetrate. So whereas ionizing radiation penetrates packaged materials, non-ionizing radiation does not. UV radiation damages the DNA, causing what we call thymine dimers. So when you have a strand of DNA, and DNA is composed of adenine, guanine, thymine, and cytosine. If there's two thymines sitting together on the same strand, they will bond together and that keeps them from being able to bond to anything else. And so that causes mutations within the DNA, and then the DNA can't be replicated properly. And then microwaves kill via heat that is generated. One form of ionizing radiation is known as irradiation. So this is used to kill microbes that are growing on food. And so here we have a bunch of, I think those are tomatoes. Ooh, 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 ooh. And what this is showing is that we can irradiate these tomatoes, and then the tomatoes will stay fresh for a longer period of time. So probably everybody in class has been to a farmer's market and has purchased or seen fresh vegetables from the farmer's markets, and those are amazing. When you get fresh fruits or vegetables, they taste so good. But if you purchase them on, say, a Saturday, and you're going to use them until, say, Thursday or Friday, they sometimes will go bad before then. And that's because these have not been irradiated. So we haven't killed any of those spoilage microbes that are sitting on the surface of the food. And so if we're not going to kill those microbes, then we need to use the food relatively fast so that it doesn't start to spoil. And this is just a cute joke about a potato. Mr. Potato Head, think about him, right? My client agreed to be irradiated. He was assured that nothing could possibly go wrong. So that's just showing that radiation can cause problems. Radiation can cause mutations, which we know already. That doesn't mean irradiation is dangerous if you eat foods that have been irradiated. We've all grown up on irradiated foods. So the next mechanism that we'll talk about for controlling growth is filtration. Filtration uses some form of a filter that has pores that keeps microbes from getting through. And so depending on the size of the pores, different types of microbes will be able to possibly get through or will not get through. So larger microbes are not going to move through, whereas really small things can. And so in this case, we have bacterial cells that are too big, but the fluid can go through. And so what we have down here is sterile fluid. HEPA filters are used in hospital ventilation systems to keep, and in laboratories, so like in BSL-4s, to keep airborne pathogens from moving around. So this is what a HEPA filter looks like. Okay. I'm going to stop here because once we come back, we'll be into the chemicals that are controlling microbes, and that's a good area. This is a good area to stop, and then I'll finish up with chemicals, okay? All right, bye.